So in this video, we are going to go through the process of protein synthesis. And in order to start with protein synthesis, we actually have to start with the messenger RNA, because that is actually how we actually have to start this whole sequence of events. So in order to understand the beginning part of the process, we have to look at how we actually form messenger RNA. And this process has a name. It is called transcription. Transcription occurs in the nucleus, and it's going to involve the creation of the messenger RNA using our DNA as a template. So what happens is the DNA partially unwinds and the strands separate, which is exactly what happened with replication. It's just we're not going to copy both sides at this time. What we're going to do is we're going to create a copy of one side, and we're only going to do the specific part of the DNA that codes for a particular protein. Now, after the DNA partially unwinds, the floating RNA nucleotides that are in the nucleus come in, and following the base pairing rule, so if they will match up. So if DNA has an A, RNA will have a U. If DNA has a C, RNA will have a G. If DNA has a G, RNA will have a C. And if DNA has a T, RNA will have an A. Now this requires an enzyme, which is called RNA polymerase. And I actually have an animation here that I'm going to show you. So with this, we are actually over, let me just make sure. Yeah, got it. Okay. We are actually over here at the University of Nebraska, and it actually is their agricultural department. All right, so these are cells, specifically plant cells, and we can tell that because of their shape. So we've got the chloroplasts in here. We also have the mitochondria, but what we are interested in here are the chromosomes. Okay. So inside the chromosomes, we have this curled up DNA. And if we look at that, you can see here the double helix then of the DNA. So we start with a particular segment. This is the segment here of DNA that we are interested in creating that protein from. Okay. So in order to understand that, we also have to look at some of the other players. So we have a promoter region, which is basically an area that tells, hey, this is where we start a gene. We have the coding region. This actually codes for the protein itself. And then we have a termination sequence. Again, this is an indication that we have ended coding the protein. We also have to have that enzyme that we talked about. That is RNA polymerase. And this is actually what helps read the DNA strand and help builds that messenger RNA strand. So the promoter, if it is on, that means that we need to copy this segment. If it is off, that means we won't copy this segment. So what happens is that the RNA polymerase actually recognizes that the promoter region is on, and it will come in and attach. And notice that when it attaches, it actually unwinds the DNA. Okay. So as it moves along, you've got nucleotides that are floating in, and you'll see that better here in a second. But then we start getting this tail coming off. That tail is actually the strand of messenger RNA as it goes. So once it enters the coding region, it starts, again, it continues that coding. And what we will see here is that we've got the red nucleotides. Those are our messenger RNA nucleotides coming in, and they are matching up on one side of the DNA. And that's what RNA polymerase helps to do. So it's unwinding the DNA at this front edge, it is rewinding the DNA at the back edge, and then what it is doing is in the middle, it's helping take those messenger RNA nucleotides and match them up just on the one side. So you can see here we're getting that free single strand of messenger RNA exiting the RNA polymerase. Now, when it reaches the termination sequence, it actually folds the messenger RNA back on itself. And that's kind of important because basically that means that this part won't be able to go through the ribosome when we are actually building the protein. So when it can't go through, it stops and we're done. Okay. So then the RNA polymerase is able to release and go, repeat this process in another section, and the messenger RNA itself heads out to the ribosome in order to make the protein. All right. So again, how does the RNA polymerase know where the gene starts and stops? Well, we saw that in the little animation there. It's because it starts always in a specific sequence called a promoter. So those promoters tell that RNA polymerase where it needs to start its process of transcription. And the termination sequence is the signal that tells the transcription to stop. All right, so now we've got this free-floating strand of messenger RNA. So before it can head out to the ribosome, though, it needs to have some editing that is done. So if you look here, if I've got my DNA sequence, and so when I make that messenger RNA strand, we actually call it the pre-mRNA strand, we get a copy of it. Now, the exons here on the DNA are the darker blue, and the introns are the green, and we get that banding pattern, so to speak. We actually don't see a band in the pre-mRNA. Before the messenger RNA leaves the nucleus, however, what happens is there's another enzyme that comes along, and what it does is it pulls out all these introns and shoves together the exons, so that way then it's only the exons that leave the nucleus and actually go to the ribosome. All right, so now that we've got this edited piece of messenger RNA, what happens next? Well, it has to be read. We know, we can't just keep it as messenger RNA. We're never going to get a protein, and it contains the DNA's instructions. So that decoding process is called translation. So if you think about it, I've transcribed, so I've kind of copied in some ways my DNA into messenger RNA, and now I need to translate it. So now I'm going to turn it into something else. So in this case, I'm going to turn that genetic code into a protein. This occurs at the ribosome, and like I said, we're translating the code into a protein. So again, this is kind of the second step in protein synthesis. So if we take a look here, transcription, which is the process of turning DNA into RNA, that takes place in the nucleus. So inside of this kind of wiffle ball looking structure here, we actually turn DNA into RNA. Okay. Translation is where we take RNA and turn it into a different type of macromolecule, so not another nucleic acid, we're turning it into a protein. This takes place in the ribosomes, or at the ribosomes in the cytoplasm. It's the same sort of idea where we take the blueprints of the master plan, we carry them out to the building site. So the DNA can't go to the building site, so its blueprints are taken by the messenger RNA to the building site, the ribosome, in order to create the protein. Now, when we get to the ribosome, we have to be able, the ribosome has to be able to read that message. And the message is carried, like I said in the previous video, in what we call a codon. So every grouping of 3 bases is called a codon. So if I have this segment here, UCG, CAC, GGU, we actually break it up into 3 three-letter words, UCG, CAC, GGU. And when I go through the process, those are going to turn into amino acids. In this case, serine, histidine, and glycine. And I'll show you how we are actually able to do that here in a minute. But what you have to understand is that different codons can represent different amino acids. So just a little bit of background here, just so you can kind of understand, what we're working with here. All proteins that we have are made up of only 20 different amino acids. So it's the pattern that those amino acids create that creates the different proteins that we have. So this gives us a very wide variety of combinations. Now, if I look at the possibilities in terms of the 4 letters that I have and the possible combinations that I have. And yes, it does matter if the C comes first or the G comes first or the A comes first. We have 64 different possible codons. And what that tells us is a couple of things. First of all, and probably the most obvious, is that some amino acids are actually coded for by more than one codon. In fact, there's some of them have 4. There's a few that have 6. There are a couple that only have 1, but most amino acids have more than one codon that codes for it. Now, one of the possible codons we have is kind of special. AUG, that codon codes for the amino acid methionine, but it also acts as the start codon for protein synthesis. So remember, RNA polymerase goes and starts at the promoter region. Well, that promoter region doesn't contain methionine. So once the ribosome actually gets to that first AUG codon, that's when it knows it needs to start making the protein because everything else was kind of the lead into that. Now, we also need to know when to end, and there's actually 3 possible stop codons. So when they get to the stop codon, there's no amino acid that codes for stop. There's no, there's no duality there. So it just stops. It knows that it's done, and it lets go of the messenger RNA, and the protein has finished its folding, and it goes off into the cytoplasm. And like we said before, some amino acids have more than one codon. So how we actually translate this is using a tool that looks sort of like this. And this is one of the tools that we will use. There is another possible tool that could be on the keystone, so I do need to get you familiar with that. But this is what we call a codon wheel. And the way we read it is from the inside. So we start with this inner ring, and then we move to the second ring, and then we move to the outer ring. So for instance, we talked about the codon AUG coding for the amino acid methionine. So if we look here, if we start with A, and then we jump out to U, and then we jump out to G, we end up with the amino acid methionine. Now remember, methionine can be used in the middle of a protein, but at the very first instance of it, that indicates that that is the start of the protein sequence itself. Let me point out a couple of others here. We have UAA, which is a stop codon. We also have UAG, which is another stop codon. Our third stop codon is UGA right here. Okay, so you can see alanine here has 4, glycine has 4. We have several of them who actually have, well, leucine has 4 here, but it also has 2 more up here. Now, we will get some more practice with this with a lab activity that we have where you guys will get a chance to actually work with the codon wheel as well as the codon table to work with your translation. So if we take a look at what actually happens inside the ribosome, we end up with, we have the messenger RNA and we have to line up the tRNA. And the tRNA has an anti-codon, so it's the base pairing rule here. So A pairs up with U, U with A, G with C. And so we know that this tRNA with this amino acid needs to come next. Okay, so it matches up with the codon, and each tRNA only carries one kind of amino acids. So UAC is only going to carry, well, let's go back here. It's actually AUG, so that's methionine, okay? So it's only going to carry methionine, and then the others will only carry their amino acid as well. So UCC, the next one that's coming along, UCC, we should have serine next. All right, so translation is the process where amino acids are assembled into the polypeptides. So just to kind of give you an overview again of protein synthesis, so we first make the messenger RNA in the nucleus. That's the process of transcription. The messenger RNA is sent out to the ribosome. It goes into the ribosome. We have the transfer RNAs that come in. match up with the messenger RNA. They each carry an amino acid, and as they move through the ribosome, so this line is just moving in this direction through the ribosome, as they move through, they detach their amino acids from the transfer RNA. So here we have this transfer RNA that has detached its amino acid. It actually has attached to the next one in the chain. And so this one will float out and it'll find another amino acid for its specific code. This one is coming in. These two are going to get attached and everything's just going to keep moving. The ribosome just kind of keeps moving down in that direction. We get to the end, so here we have a bunch of different amino acids in the polypeptide, and we've reached the termination or the stop codon, and so the ribosome is going to finish folding this protein and assembling it, and the messenger RNA will actually fall apart. The tRNA will go pick up some more loose amino acids, and the process will start again. What you have to remember is that the protein itself, what it does, how it looks, what its shape is determined by the order of amino acids. Those amino acids are given by the messenger RNA sequence, and that messenger RNA sequence is actually determined by the DNA. All right, let's get this. I will actually put this video up as a separate link on there. We won't embed it here just because the sound and everything won't come through. All right, so what we've got to kind of keep in mind is that DNA can replicate itself. Okay, and that's the process of mitosis, for instance, that we need to do that, or meiosis. But the central dogma of biology, in other words, the main purpose of molecular biology, is DNA turning into RNA, turning into proteins. That is kind of the central focus now, and part of what we call the molecular biology revolution. The process of DNA turning into RNA is transcription, and the process of RNA turning into a protein is translation.